So my name is Henry Rodriguez, and I currently serve as the director of the Office of Cancer Clinical Proteomics Research at the National Cancer Institute. And one of the things that I'd like to do first and foremost is to thank the tremendous folks behind the scenes that actually brings this webinar, sort of a slash podcast, as I like to say, to those that are online today. And that is the US HUPO organization and also a subcommittee within US HUPO. The chair of that happens to be Andy Tao, who's at Purdue University. Also part of the committee is Olga Vitek at Northeastern University. There's Peter Nems at the University of Maryland here in College Park, and also Stephanie Colonna at the University of Illinois at Chicago, and also myself. Huge thanks, I have to say, also goes to Jennifer Jones at US HUPO, which pretty much controls all the logistics behind the scenes, and to the current president of the US HUPO, which sort of blessed this idea of doing these webinars, and that is Jennifer Van Eyck at Cedars-Sinai Medical Center. So for this month, it is a great honor for me to actually be conducting a webinar with an individual that I've always considered to be not just a scientific colleague, but really a dear friend that I've now known for approximately about 20 years, I'd say, and that is Dr. Josh LaBaer. Is that a fair assessment, Josh? We've known each other that long?
I think it's fair. I mean, we have shared $1 tacos together.
That is true. That is true. So Josh, you know, we have to get serious here, because one of the things that I plan to do for the audience is I have known you first on a professional level, and then I got to know you on a personal level. And it's been a phenomenal journey for me, quite frankly. And it's one that I think what I'd like to do for today, for this webinar, is really to kind of present you as an individual, both at a professional, and also kind of delve into a little bit of your personal life and what has driven yourself to gone after the sorts of path that you've taken, and lay that out for the audience so they could get a good sense on, in terms of a scientific career, whether it be in medicine or just pure science, and how to balance that. Do you think that's something that you'd like to participate in, Josh? And are you game for it?
Looking forward to it.
Okay, great. Okay, so Josh, so now we're going to get serious. So here goes the hard questions. So let's talk about your education.
Yeah.
So you're born in upstate New York. Ultimately, you moved to Arizona as a child, and you're essentially raised there.
Yep.
All your schooling through high school.
Yep.
Next thing, you decide to pack your bag. You yourself now. So you're a young buck, and you end up moving even more west in Arizona. So now you end up going to California this time, a state that I also lived in. And you moved specifically to Northern California. Yep. So there you attended some of the state's most prestigious institutions. First, you went to UC Berkeley.
Yep.
You obtained your Bachelor of Science degree in nutrition. Then you ended up going not too far away, but you went to University of California, San Francisco, where you earned your dual degree, an MD, PhD. Your PhD was also quite unique. If I get my facts here straight, it was sort of a combination of biochemistry with biophysics. So is it fair to say, Josh, can we characterize you early on as being a total bookworm?
Yeah, I mean, you know, it's funny. I think back to college, and, you know, I must have done well because I managed to get into medical school and all that stuff. But I don't remember studying that much. I remember all the fun I had at Berkeley. So, you know, it's funny how memory does that to you, because, you know, I remember taking trips into San Francisco and going out to the wharf and doing hikes and doing bike rides, and yet somewhere in there I must have done a lot of studying. So, but yeah, I mean, obviously I kind of hunkered down and went through school kind of straight through.
Very good. Okay, so basically what I heard is that you have a unique talent where I did not have time to do all these little excursions. I had to be on those books yourself. So that's actually really good. But, you know, one of the questions that I tend to get a lot, either from students or professionals that are actually trying to decide which academic path in terms of the degree to pursue.
Yeah.
Why did you actually, it is interesting because you do have that dual degree. Why did you choose an MD coupled with a PhD as opposed to strictly going after, let's say, the MD or the PhD in isolation?
Right. So, you know, I grew up in a sort of a typical middle class kind of environment, and I had a very limited view of what the future meant, and I was thinking either I wanted to be either a lawyer or a doctor or something like that when I started college. And then early in college, like maybe my sophomore year, I took an honors organic chemistry course. And what made it an honors course was instead of doing the sort of cookbook experiments in the lab that are guaranteed to succeed, we had this sort of 10-stage complex synthesis. And the professor explained to us, this was Hank Rappaport, for those of you who know Berkeley, explained to us that no one had ever done this synthesis before. And that the idea that we were doing something that no one had ever done before kind of tripped something in me, like, wow, that's kind of cool, doing something for the first time. And that made me think, boy, I should get involved in research a little bit. And so I started volunteering in a lab to do some research. And this whole concept of, sort of exploring the unknown in the context of research bit me hard. I just got really excited about that idea. So at that point, I realized I probably didn't only want an MD. I probably wanted to do a PhD. And then when I heard that there were these programs where you could do both, that seemed very cool to me. And it didn't hurt that they had funding attached to them so that you could go through medical school with funding. Although financially, it's much better just to go to medical school. But nonetheless, I think for what I wanted to do, it was pretty exciting. And so that's what kind of drove me to the MD PhD approach.
Okay. So you talked about your interest there for the MD PhD and the lab work that you were doing. So while you're actually obtaining your MD PhD, you actually did a series of summer research rotations in several laboratories out in the Bay Area. One of them in 1982, you ended up doing a summer research rotation, actually in the laboratory of Michael Bishop.
Yeah.
Now, what's interesting is that this is the summer of 82. So 7 years later, 1989, so Mike Bishop gets awarded the Nobel Prize in physiology or medicine. So do you think that there might be some aspects of the work that you did in that summer in any way, shape, or form that might have contributed to the award of that Nobel Prize?
Not. Almost certainly not. I will say I am deeply indebted to Mike Bishop because he, when I joined that lab, I had done a nutrition undergraduate degree, which was great. It was very much involved in isolating compounds from vegetables that were anti-cancer and their behavior and all that stuff. But there was no molecular biology involved. And when I joined Mike's lab, he was like my first rotation. And when I started having conversations with him, he realized that I had no clue about molecular biology at all. And he sat me down and said, I'm going to give you some articles to read and we're going to do them together and we're going to get you up to speed. And he did that. It was really amazing. I mean, someone of his stature, even then, even before his prize. And he gave me a series of articles to read and he kind of coached me. And so I benefited way more from that rotation than he ever benefited from my work. But it was a great experience.
Well, I think you're being a little bit modest. So let me kind of delve into that a little bit more. So later on, in 2003, Mike Bishop, he's being awarded a lot more sort of awards and accolades throughout his career. And one of them he now gets is the National Medal of Science. Now, this is extremely prestigious because that gets ultimately bestowed by the President of the United States. Yeah. Do you think, again, and I'm going to go back and I'm going to put on it, that summer of 82 might have contributed to that award? And I'm trying to help you here.
I had a great time in that lab, but I don't think that I could take any credit for what, the astonishing work that both Mike and Harold Barmas achieved in their work. And in those days, they had a joint lab. They ran their labs together. And it was a really exciting place. I mean, they were doing all these southern blots to look for oncogenes in cells. So it was pretty cool stuff.
Okay, so now following your MD PhD, at this point, quite interesting, you decided to move east at this point. So you moved to Boston and you basically went there for your postdoctoral training. There's a combination. You did your internships with your residency. That was in internal medicine and medical oncology, and then followed by your fellowships. Now, I believe it's during this time, maybe second or third year into your training out there, your mother, which was an English teacher, passed away from breast cancer. Yeah. Now, that cancer was initially detected through imaging, through mammography. Yeah. Ultimately, she had surgery. It showed that it was advanced. It did spread to a couple of the lymph nodes. And then 3 years later, it moved to the brain, where despite radiation treatment, she ultimately succumbed to cancer at a young age of 64. What role did that life experience and very early on in your training now in research coupled with medical oncology, I mean, like how did that play on your approach as it relates to tackling cancer?
Right. It impacts you in a lot of ways. So I was already training to be an oncologist at that point. And, when something like that happens to you personally, all of a sudden, your whole perspective on the disease and to some extent, the powerlessness that we have with regards to these diseases really impacts you. So, she was across the country. She was in California while I was in Boston. When her tumor was first found, she had 17 positive lymph nodes. So we knew from the beginning that this was an aggressive disease. We got her on a clinical trial. They gave her high dose chemo, probably cured her body of the cancer, but as you know, the brain is kind of a protected site where a sanctuary site where chemo doesn't always reach. And so not surprisingly, a year or two later, it showed up there. And, you realize that you can do all the things you know how to do. And despite getting her enrolled in a trial at UCSF and all that stuff, the cancer is, it's a bad actor. It certainly influenced me to realize that the best shot we have at treating cancer is early detection. And so that was one of the things that motivated me in my career to focus on that aspect of the disease. It also made me realize how important it was to do research in cancer because it is such a challenging disease and one that we still face today as a major challenge. So, and of course, a lot of the work that we did in my lab was around breast cancer, in part because of that whole experience.
So that is interesting because your career really has focused on the early detection, and it is intriguing knowing if that would have been detected very early on. Obviously, the outcomes are always greatly improved.
Yeah.
So that clearly played a very significant role. So now, following your training in Boston, obviously you got the bug bite, and you started to stay in Beantown, right? So in 1990. Now you're at Harvard Medical School. You end up doing your clinical fellow in medicine, and subsequently that transition into an instructor in medicine at Harvard Med. So for those of us, I being one of them, to be very candid here, what is exactly the difference between a clinical fellow and an instructor at Harvard Med?
Oh boy, to be honest, I don't really know all the details of all that stuff. I think instructor, I believe, is a faculty designation of some form, whereas fellow is still a trainee. Harvard is a complex place that I don't fully understand to this day. So there are many of you probably who know it better than I do. But yeah, I kind of finished up my fellowship and moved on to this instructor position in part because, my postdoctoral advisor, Ed Harlow, had brought me this opportunity to start a center, a little mini institute, if you will, within the medical school around this new technology that we were playing around with. And it just seemed like a cool thing to do. So I went that direction, and the instructorship came kind of with that package.
So let's talk about a little bit more about that little venture that you just talked about. So
Yeah.
now you're about 10 years at Harvard Med.
Yep.
And you go from a fellow. Now ultimately you become an instructor.
Yep.
I mean, in a way you could kind of say, wow, so like I've kind of reached it. You could do one of those little brush on the shoulders. But now around 1999, you decide to basically take that offer, and you now are the founding director. That little institute, by the way, if folks aren't familiar with it, is the Harvard Institute of Proteomics at Harvard Med. So what was the vision, the light bulb that literally went in your head, that went off to actually create the Harvard Institute of Proteomics?
Yeah, so this work began when I was still a postdoc. We had been contacted. When I say we, I mean really Ed Harlow and the Harlow Lab, by this group at Life Technologies at that time, which eventually, in many years later, became Thermo Fisher, and then it was called Life Technologies. They had come up with this way of recombinational cloning, where you could transfer and insert in frame from one plasmid to another in a simple overnight step with near 100% efficiency based on this strategic use of selectable markers and so on. And it had occurred to us that this was potentially a game changer. It meant that you could build a library of genes in a master vector, and then in using automation, transfer them simultaneously into any protein expression vector for high throughput experimentation. So it allowed you in practical terms to screen hundreds, if not eventually thousands of different proteins in effectively a limitless set of contexts. So, the idea of this institute was, could we pioneer that approach? Could we start building libraries of genes, put them in vectors, and then develop ways of transferring them so that we could do high throughput experimentation? And of course, a fundamental component of that was whatever we made, we shared. So any plasmids we ever made, we gave away to anyone who wanted them, and we do that to this day. My lab now in Arizona has around 18,000 full-length human genes in these vectors, so we have nearly the complete set, and we still share them to anyone who wants them. So, so that was the idea. Get this technology up and running, and there was a lot of sort of different aspects to getting the program off the ground. You had to have some handle on databases. You had to have some handle on automation. You had to have a good handle on molecular biology. You had to, be able to think from it from multidimensional approach to make it work. And I like that kind of problem. So that's kind of why I started that whole plan.
And then you talked about giving all the information or essentially giving your SOPs, the clones, out to the public. So was that something that was, I don't want to use the word mandated, but was that sort of the mantra being academic, or was just simply a philosophy that you felt, as opposed to taking a commercialization route, this is something that would empower the rest of the global world if they had access to such a resource?
Right. Well, so to be fair, Ed Harlow has always been a tremendously open and collaborative individual, and the style of his laboratory that he trained us in was, you share everything. That's how academics work. I really appreciate that philosophy. I agree with it 100%. And so when we started the Institute, that was just a core value that we adopted from the beginning. I mean, there was never a question about that. Whatever we made, we gave away. And we, not just sharing the SOPs, but the actual clones themselves, we shipped them. To date, our group here in Arizona has shipped, I think, over 400,000 plasmids all over the planet. Hundreds of countries and every state in the union.
Okay, so you talked about a little bit about Arizona. So let's kind of delve a little bit into that. So now you're about 20 years at Harvard. All of a sudden, in 2009, you decide to move to Arizona State University. What was going through your mind to literally make you want to say in your head, I'm going to leave Harvard and I'm going to go to Arizona State University. And you know what? Once again, I'm going to start from scratch.
It's funny. It wasn't all of a sudden. It took a little while. It began with an email from Lee Hartwell, who invited me to a meeting here in Arizona very cleverly in February. So anyone who's lived in Boston knows that, getting out of Boston in February is not a bad choice. And certainly an invitation from Lee Hartwell, who, was the founder of the field of cell cycle, which is what I did my postdoc in. You're not going to say no to that. So I came to a meeting here, and while presenting something here at Arizona State, at that meeting, folks here had approached me about taking a potential position in the Biodesign Institute. And at the time, I was not looking to do any changes. I was more than happy at Harvard. I was having a good old time. But since I did, as you pointed out earlier, have family here, and there was a family event coming up later that spring, they offered to fly me to my family event if I spent a day at ASU. So I did. And when I got a sense of what biodesign was about, I was really intrigued. So this is a whole institute set up for translational research. So not research for the sake of research, but research for the sake of solving problems, which I liked. And it was this institute that really understood scale, doing things at a large scale, and, created to kind of develop people who had, multidimensional teams that had, expertise in lots of different areas, and which again, fed well into the kind of work that I like to do. So, over the course of that year, I had a number of conversations and visited a couple of times and decided that this was an opportunity to really build something big. And so that's what got me out here. And that's played out. I mean, we have a large team now and really enjoying, running the Institute now.
And then are these conversations that took place there on campus? Did you go up to, I don't know, Camelback Mountain, crack open a bottle of wine, and you say, yes, this is a place that I could be at?
Well, certainly the visit during February, and remembering that, in February, we're walking around in shirt sleeves because it's just gorgeous here in February. That certainly made me realize that I wouldn't miss the New England winters. Summertime's a little warm, I'll be honest, but it's only 3 months, 9 months of the year. It's really lovely here. And certainly did a few hikes, took in the valley. I mean, this is a great place to live if you like the outdoors, because you can spend most of the year outside.
So now you've been at Arizona State University, what, ballpark-ish 12 years.
Yeah.
And one of the things that's quite interesting is that commercial success has, in a way, come your way. One of the things you ended up doing was you ended up developing a blood test for all types of cancers. Guess what? In this case, for breast cancer. And it's interesting because it measures antibodies in individuals and it treats them as a surrogate actually for the cancer. Yeah. So how did you know that measuring antibodies as opposed to going after the actual antigen in individuals could potentially work as a biomarker, and in this case for breast cancer?
So that work began even while we were still at Harvard. I had early on forged a collaboration with Karen Anderson, who also has since moved here to Arizona. She was at the Dana-Farber, also a fellow, as I was. And it dates back to work that Lloyd Old and others have done on this concept of autoantibodies. The concept here is that cancers obviously produce aberrant proteins. At some point, these proteins escape to the bloodstream where they trigger antibodies. And patients who have cancer will develop these antibodies compared to healthy individuals who typically don't. And so the presence of the antibodies betrays the presence of the cancer. We like that approach because even if the antigens themselves are no longer around the bloodstream, the antibodies persist. So the antigens may be released early on in the cancer. They may go away, but the antibodies are still around. And again, the whole goal here is to detect cancer early. So you want to be able to detect cancer when it's small, when it's millimeters in size, not when it's centimeters in size. So antibodies in some ways act as an amplifier for that signal. The trick is to know what antibodies to look for, and that's where the protein array technology was ideal. In Lloyd Old's days, they would do phage display, but the problem with phage display is it was based on cDNA libraries, which tend to favor abundant proteins. The advantage of a protein array is that every protein gets its shot because you just put all the proteins on the array and you're testing all of them side by side.
So what's interesting there is that ultimately you end up licensing this out to a diagnostic company, to what now I believe is the first CLIA certified blood test for breast cancer. So what lessons did you learn in terms of that approach, going from using the academic setting for your discovery and eventually handing it off now, which is like your little baby that you created, to a company for the commercialization side, as opposed to you yourself, let's say, spinning it off into a small corporation? Why create that separation, and what did you learn from that?
Right. Well, a number of lessons learned there. So, our decision to license it was based on the notion that to get a diagnostic into the market is a pretty involved process. You've got to do large scale clinical trials that get, FDA clearance ultimately, and you've got to raise funds to do all that stuff. And that's certainly not my expertise, is, going out and chasing down all those samples and doing the large trials. Our expertise is in the molecular biology and the discovery part. So it was a little bit easier to go to a company that had experience in those areas. So that part, and I still think that in many cases that may be a simpler solution. Certainly for those who have the stomach for it and really want to build it from ground up to do it, hats off to those folks. But for me, it was a little bit easier to hand it off to somebody who knew what they were doing. That said, it's a rocky road. The diagnostic space is a challenging space. It doesn't have the big upside that therapeutics do. So it's a little bit harder to come up with the funding, and yet it's still expensive to develop because of the need to do these clinical trials. So the company that initially took on the license for Videssa, they ran some really nice clinical trials. They got some really great data, but they were a company, and like any company, they had issues, and their issues had nothing to do with the science. They had other administrative issues that led to them losing their funding. And so the test went away for a while. And now since then, the test has been bought by another company that has the funding and what it needs to kind of carry it through. And so Totus Pharmaceuticals is now going to pick it up and distribute it this year. So, you know, and if you talk to people who have developed other diagnostic tests, you'll get similar stories. I didn't realize it at the time, but some of the diagnostics on the market today have been in the works for sometimes 2 decades to get them from their initial discoveries all the way to their diagnostic tests, which then later go on to become blockbusters. So it's a long road. Diagnostics are just as therapeutics are.
So on a related topic, when it comes to commercial success, I tried my best to go through the USPTO and to look at patents. And an interesting sort of a trend came out of that. So you were at Harvard 20 years. I was able to at least track down 3 patents during that time period.
Yeah.
You moved to Arizona, you've been there only 11 years, and there's 12 patents that I'm able to find with your name affiliated with it. So is there something, Josh, in the Arizona desert or the underground aquifers that makes it conducive to filing patents? And quite frankly, if it is something in Arizona, are there homes out there for sale that you could recommend to me so I could move?
Yeah, it's interesting. Well, first of all, we should be fair that half of my time in Boston was in training. So you know, the opportunity to do patenting really didn't happen a lot until the second half. But I would also say that ASU is a university that focuses on innovation, and they value very highly filing intellectual property. And so although I know Harvard values it highly as well, Harvard is a lot more selective about what it's willing to take to the patent process. ASU is aggressive about it, and they look for new technologies and new ideas, and they really push that forward because innovation is kind of the lifeblood of our university. So in that sense, it was very easy to start filing intellectual property when I got here.
Is it a gated process, Josh? I mean, do you go from provisionals in terms of volume and ultimately out of those provisionals, which you have a certain window, then you determine which one I will do a full filing?
It's very similar to everywhere else. Everybody reports their inventions. Not every reported invention gets taken through the patent process. I think the technology office looks at each thing and decides, you know, is there potential for this or not? If they believe that there's potential, then they contact the inventors and start going through the process of filing the provisionals. And then ultimately, someone's going to come through at the end and pay for the patenting. And so the goal would be to get things licensed. And if no one picks up the license, then they may not keep the thing going.
Okay, so clearly you've done a lot of research, but you can't do research without funding. So I tried to do a tally. I broke out the calculator, and one of the things I was able to find is throughout your career, you've tallied up close to $150 million total dollars in terms of funding. Now, to be very fair, that is a combination. I mean, it's a whopping number by any stretch of anyone's imagination, but it's a combination of you serving as a principal investigator, co-principal investigator, or even co-investigator. So can you speak to people today on the role that collaboration has actually played in that success? Or quite frankly, is there some secret sauce or a template that you can actually share with the general public?
Collaboration is the key. It's without a doubt the key. It's the way that science needs to happen today. And it has been the source. I think if you looked at any one of those grants, you would see that there were collaborators on them. I don't think I have any, or maybe just a handful of grants that are just our laboratory. Almost always we're collaborating. And I think that's the key to success is realizing you're good at what you're good at, but that real science needs people with different disciplines and different approaches that come together to make big things happen. And so, my training has always been around collaboration, and I've featured that heavily in everything I've done. And so I think that's the key, is you seek out the best people at whatever elements you need to build out the program, and then build the grant around that. And of course, every successful grant I've ever had began at a whiteboard. You get people around the whiteboard and you put up your ideas, and then the first question you ask is, okay, what's wrong with this? And you beat it up, and you figure out what's wrong with it, and then you figure out who you need to bring into the team to make it a solid approach that will survive.
So at Arizona State University, I've noticed you wear lots of hats, titles. So let me kind of run through them. Executive Director, Biodesign Institute. Director, Virginia Piper Biodesign Center for Personalized Diagnostics. You also serve as the Robert Dalton Chair for Cancer Research. You're also the Virginia Piper Chair of Personalized Medicine, and let's not forget, you're a professor of the School of Molecular Sciences. Do any of these come first? And if not, how do you balance them?
Well, honestly, the one that takes the most effort is being the professor. Without a doubt, that's the hardest one, right? Kind of keeping your lab running and keeping the science moving and making sure that you're making progress. That takes up a huge amount of time. I'm enjoying running the institute. The Biodesign Institute's a just phenomenal place. We have just outstanding faculty here. Everybody creative, everybody doing really cool science, and the opportunity to kind of build that institute and grow it is huge for me. Arizona State is still in growth mode, so we're constantly hiring new people to start new centers and build new disciplines, and there's nothing more fun than doing that. So I think between those two, I keep pretty busy for sure.
So you talked about a lot of fun, and you talked about the professorship. One of the lot of funs that I know a lot of us has as we get up there within our careers is training the next generation of scientists or physicians that could actually discover more things than we could even imagine as technologies evolve. So throughout this point, you've trained now close to 40 students thus far. So what are the challenges, if any, have you faced while training and mentoring students?
Oh, wow. I love training students. Every single one of them is different. They all have their unique styles. And part, I guess, of the challenge, of course, is adapting to those styles to some extent so that you can help the student succeed in the best way that he or she needs to succeed. Some of them are just, get out of their way. They'll succeed if you just don't block them. And then others you have to really kind of nurse along because they're, for whatever reason, they need that attention. So, I think another challenge, of course, for me is that my schedule is really busy. So I have to make sure I make time to interact with the students. I usually at least meet with them every other week, if not every week. And in some projects, if they're really active, more often than that. But yeah, it does, training people is, of course, what we're here for. That's what universities are meant to do.
Do you have a favorite piece of advice then for students that you would like to share for those that potentially want to train in your lab or any other laboratory for that matter?
Well, I guess I would, for my lab, I would say, you definitely need to be self-motivated. I think graduate school is very different from undergraduate. In undergraduate, the courses are laid out for you and you're told what to do. In graduate school, you're not going to be just given a pipette and said, just go to it. But if you don't drive yourself, the process just won't happen. It is a little bit more of a self-defined journey, if you will. So I would, advise them to make sure that they kind of keep moving on their journey.
So now let's focus on some current events. So the pandemic. So COVID-19 hits in early 2020. And quite remarkably, I think one of the first things that you did was to mobilize Arizona State University and make it a center in the state's race, ultimately to contain COVID-19. So specifically, you actually helped to develop and implement diagnostic screening tests for the public there. So was it easy making this transition? I mean, also, did your current research programs or training that you've done throughout your career actually help on getting that off the ground?
Yeah, it was both easy and extremely challenging. So without a doubt, my background was perfectly suited to developing a diagnostic. My whole field of research had been in the diagnostic space. And so I was familiar with a lot of the issues that need to happen if you're going to develop a diagnostic. And that made it a little bit easier for me to have the broad vision, if you will, of what needed to go. But at the same time, it was extraordinarily challenging because the tools we needed were not there at the time. So, we developed a qPCR test for COVID-19, for SARS-CoV-2, and we initially did that with nasopharyngeal swabs, although later we completely transitioned to saliva. But when I first thought about doing this, a group of us started meeting every day of the week at 8 a.m. to start discussing it. And we contacted the hospitals to see, where we could get swabs to start collecting samples. And it was clear at that point that there were probably no more than 1,000 swabs in all of the state of Arizona. Hospitals would report that they had 200 swabs, collection kits in their entire hospital, and they were going to keep 100 for their staff. And so we had to figure out how we could make those kits ourselves. And sourcing that stuff back in February of 2020 was just a nightmare. And so we had a person all he did was make phone calls to see what he could get in terms of supplies. And then, we had to figure out what the chemistry we would use. We fortunately had equipment already in place because of a previous study we had done for an agency called BARDA, where we were detecting gene expression in response to radiation. And so we had, the qPCR instruments, we had the automation. And so we were able to develop that early, but then as time went on, we had to scale because we realized the state needed help. And so that's when we switched over to saliva, because it's a lot easier to do a high throughput collection in saliva than it is to do nasopharyngeal swabs. And then we had to get a lot more instrumentation in place and really make the automation a lot fancier than it was originally, including cappers and decappers and the whole process.
So that's interesting. You talked about the saliva one. So, in a way, I think it's good for the audience to know. I mean, that was not an easy feat. And in fact, what's quite nice about it, I believe that's the state's very first saliva-based test for COVID-19.
I think we may have even had the first public test in the country.
Yeah,
I know
so
tests are already available, but in terms of public
from
testing,
all of us who take frequently nasal swabs, thank you.
Yeah.
So can you speak though? I mean, you spoke a little bit to the development of that test. You actually see it, Josh, as a companion, or do you see it as a replacement? In this case, for the qPCR swab, nasal swab.
Oh, I think it works as well as the NP swabs. We used it as a base form of identification. Routinely, in part because of who and what we are, we were testing, because we're not a hospital, right? So we're never testing people who are sick. People who are sick go to their doctors to get tested. We were testing largely an asymptomatic population and routinely found the virus even in asymptomatics. So it worked really well for that. The key was, working out the chemistry, and one of the challenges, of course, is the viscosity, because what we learned early on was the best way to collect saliva for us was just collect it neat, if you will. Putting buffers in it just diluted it, and it was quite stable, surprisingly stable. We did a whole temperature series, and we could testing out to 96 hours, even at room temperature. We had almost no diversion in the CT scores, so we could get very good results even at long periods of time. And so that, our big innovation was using a drinking straw. I know it sounds silly, but what we discovered being in Arizona was, there were some tobacco chewers out there and other things going on, and we would get not saliva. We would get phlegm, we would get snot, we would get other things. And if people are forced to get it down through a drinking straw, what you get at the end of that straw is saliva. And that's how that was our quality control step that made sure that the samples would work. And then of course we had to develop the automation that could handle that viscosity and and work out the right chemistries and all that stuff. But once we had that working, we were able to scale it to, we were routinely running 10,000 samples a day.
Hmm. So this work actually has garnished you some quite, your name is dropped in the press. I mean, you're covered by the local media all throughout Arizona. The press covers you out there, including I was able to find going through USA Today, your name is actually in several of articles in USA Today. There's one in May, one of 2020, one in June, July, August, and in September of last calendar year. So what are the chances you think that we may be seeing you on CNN in the not so distant future?
Not high. I'm a modest sized fish in a very small pond. So yeah, yeah. I mean, I think, part of all that was that, Arizona managed to kind of lead the world at least twice in terms of the density of new cases. So I think, as the COVID wave passed across the country, it seemed to end in Arizona, where when everybody else was through, we would get these really high runs of counts. And so I'm kind of a local person here. So that's how I ended up in the press, I think.
So the recent work in the pandemic and also your prior career, you've earned a whole series of awards and accolades. I'm not going to riddle through all of them, but one of them actually stands out. So in 2020, you received the Arizona Governor's Innovation of the Year Award, and that's related to ASU's Biodesign Institute's response to COVID-19. Did you get to meet Josh, the governor of Arizona? I mean, was there like a dinner banquet involved? And if so, what was it like?
No, it was all done, a virtual event like everything else was at that time of year. This last year, everything was done online. So no, I didn't get to meet the governor for that. I did.
Is there any possibility that still might come down the road? I mean,
never know.
Are there pictures you could share down the road? Because that would be very fascinating.
Certainly met on a number of occasions. Kara Christ,
a little invitation coming my way. I'm just dropping things here as suggestions. That's all.
You're hot to meet the Arizona governor, huh? Yeah.
It's a free dinner.
Yeah, there you go. Yeah.
Okay, so speaking of quarantine, let's get a little bit more like a light topic here. Let's talk about some of the hobbies that you've had a chance now to brush up while a lot of us have been on lockdown.
Yeah.
So one of your hobbies that I know is photography.
Yeah.
So where does that interest actually come from? And do you consider yourself to be a point and shoot hobbyist, or are you talking an amateur, semi-pro photographer, talking digital full frame cameras?
Well, I do have a digital full frame camera, but I'm very much an amateur. I think photography, for those of us who have no talent at drawing, and I have zero artistic ability with my hands. The nice thing about photography is that it's just technical enough that I can use that side of my brain, the left side of my brain, to improve the quality of the image, and yet find ways to try to express myself with images. So I do a little bit of landscape photography when I can get out and about, and then of course I do I don't know if you'd call it journalistic style when I'm taking pictures of my kids or whatnot.
So that's interesting. Do you then enjoy taking pictures more of landscapes, or do you enjoy taking pictures more of individuals?
I like both, actually. The one downside of photography is you're always standing there with a whole handful of gear, and your family's kind of like, come on, let's go. Stop taking the pictures. And so what's sometimes fun is going on my own hikes and then just taking pictures on my own, so I'm not holding people up. But if you're in exotic places, you're often with family or whatever, and then everybody's waiting for you. So you got to get it done quick.
Are you one of those like me then, where you look at the family albums on vacations and typically there's one missing, and that's you either taking the picture because it's too long of a time to prop it, put on the timer, run back to like, Jesus.
I'm in almost none of the family vacation trips. Yeah. Unless someone took me on their phone.
Yeah, exactly. Cooking. Let's talk about cooking. So here I know you are more than an avid cooker. I mean, you and I
both, right? We both cook, right?
We both share a lot of recipes
for each other,
behind the scenes. That is true.
Yeah.
That is true. But what draws you to cooking, where does that passion come from?
So that started when I was a teenager, when I became vegetarian for about a decade. And so when I went to Berkeley, Berkeley has one of the housing options in Berkeley is they're called cooperatives, and they're student-owned buildings, and they're much less expensive, a third of the price of the dorms. And for me at that time, that was helpful. And you, instead of paying the higher fees, you guarantee that you're going to work 5 hours a week for the house. You do something. And one of my work shift was the Friday night chef. So I was the cook for 55 people on Friday nights. And so I started to take it seriously and became pretty good at it. And so I'm no longer a vegetarian, but the interest in cooking started then and has persisted since.
Did you perfect any new recipes during the quarantine? I mean, bread seems to be the most popular one.
Yeah, yeah, but I shouldn't be eating a lot of bread, to be honest. So, but I've been doing a lot of cooking during. In fact, pretty much the whole year. My one joy outlet was going out on Saturday mornings to the farmers market and picking out produce and then cooking on the weekends. I don't really cook. I reheat on the weekdays, but
Right.
I love cooking on the weekends.
So there's another interesting hobby that we were able to find out. I mean, because we have our sources all throughout the world and we basically dug a lot. One of them, it seems that you have a little hidden gem, and that is you are a music concert videographer. So in 2018, we were able to dig up that the School of Rock Gilbert Band actually performed there in Arizona at the Wasted Grain in Scottsdale. Sweet Child of Mine. And the credits for the video production, the editing goes to a Joshua LaBear. So is this like a new title that we should see on your busy roster of already numerous titles?
Yeah, I was dabbling. Let's just say, most of my photography is still photography, but I had some friends who were playing in rock bands, and they asked me to take some video, and I figured out a way to do a 2-camera system. So I put one camera on a tripod and just filmed the whole song, and then with the other camera, I would wander around and take close-ups, and then figured out how to play with Adobe Premiere Pro to edit them together. So yeah, it was kind of fun.
Will there be other sorts of rock bands, concerts that are kind of in the works, or was that like a one-off experiment?
That was a one-off. That's a lot of work, that stuff. Yeah.
Okay, so let me do some closing thoughts, Josh, and then I'll kind of go, there's a couple of questions here. So are you then optimistic about how our understanding of cancer has progressed, let's say, in the past 20 years? And what do you think still needs to be done?
Yeah. So I am optimistic, actually. I think, if you look at statistics, we're getting better. The numbers are coming down for a number of cancers now. And I think this, the combination of more and more improved targeted therapies combined with all the new immunotherapies that are being developed, I think I think the future is going to be good. I think we're all realizing that every single cancer is different, and that even within a single cancer type, person to person, it may be different depending on which pathway. But we have a pretty good understanding now of most of the pathways the biochemical pathways that lead to cancer. And I think it's just a matter of time before people come up with better and better ways at targeting those pathways to try to inhibit those cells and give the immune system the time it needs to make a difference. So I do think there's a lot of hope there.
Okay, so in the couple of minutes we have left, let me kind of go through a couple of the questions here, Josh.
Okay.
So one of them, actually, so this one is, how do you envision, in this case, using proteomics now for the application to for the diagnosis of cancer, but actually through liquid biopsies, exosomes?
Well, I think exosomes look like a really promising tool for identifying cancer. And I think, we've seen a lot of papers that show just remarkable results. And sometimes those results look a little too remarkable to me. But what that means to me is not that they're totally false. It just means that This clearly a powerful technology. We just have to hone the statistics right and get the numbers correct. But it does look like, these, what we used to think of was just junk that came off of cells are really, UPS packages that are carrying specific information in them, including, targeting information. And if we can learn how to read that information, we can learn a lot about not only what cancers might be present, but also, maybe even subtypes of those cancers or unique elements of those cancers that may help us better treat that individual. So I think we need to see where that goes, but it's very promising.
Oh, I agree. I mean, as technology evolves, I mean, you can't have advances without technology development. It's a
Right.
beautiful 360 pattern, which is what you get. So there's another question. This one's somewhat related to the advice that you're giving to potential students that you're seeking. So this one is, are there any suggestions or recommendations that you'd give in this case to young grad students that are engaged specifically in cancer research while at minority serving institutions? Um, any advice there, or is it sort of the same advice you could give at any institution?
Well, I think the general advice I would give is, obviously follow your passion, do work in areas that interest you because you're going to be more engaged if you're following your passion. And of course, be self-directed, drive yourself to move your project forward. And then I would also suggest strongly that you get out and go to meetings. I think that's really key. I think a big part of being a student is getting exposed to other scientists and building relationships. And that may be even more true for minority serving institutions because if they're not getting the constant flow of people through them the way you might get if you were at MIT or UCSF or Stanford or whatnot, it's important to get out and go to those meetings and meet people so that people know who you are and that you get practice at presenting your science. Go to
And
HUPO.
there's okay, and no, I agree. The key is to go out. I mean, I don't need to just socialize, but the reality is networking is very key. And I think building very strong collaborative and friendships and trust for others is how you grow as scientifically a career and as a person. So Josh, there's one more that I like to cover here. It's sort of tackled the aspects of what you did for the COVID-19. So this one kind of basically is asking, so that experience that you had on taking your science of what you did at Harvard Institute of Proteomics and starting up your laboratory at ASU, ultimately now quickly doing not as much as a 180. It sounds like you really did a pivot with lessons learned of experience of your background and applying it to a new area of direction. How do you see that science going forward into potential new avenues that you could branch into?
Right. Well, we're pretty excited. My history is, as you know, cancer, obviously, but this infectious disease approach that involves immunology, obviously, the same way that our cancer research has. We're now very excited about studying serology in the context of SARS-CoV-2 and COVID-19. We're part of the SeroNet program, which is an NCI-sponsored program, as you know, and we're using a derivative of the technology that led to our protein arrays, but it's kind of a more solution-based version to develop a test for, multi-analyte test for looking at not only SARS-CoV-2, but other related viruses to see how they may relate to each other in terms of infection, severity, and all the rest of that stuff. So we're pretty engaged now in that process, and it's pretty exciting. I think, opportunities to really have an impact, not only I think on SARS-CoV-2, but also on whatever the next thing might be. So, because there will be another one at some point, and we want to have the tools ready for that one when it comes.
Okay, great. Well, Josh, we've come up to the hour. On behalf of myself, my colleagues within US HUPO, and I think for everybody that participated on today's webinar, we'd like to give a big thank you for you for being a good sport, for taking time out of your calendar to do this interview.
Oh, well, and thank you for all your preparation. Really impressed.
We do our best to do our homework, but Josh, again,
but you got the goods.
Well, we'll be sharing a lot more recipes very soon. And I do have a very nice one that's coming up. This one's in the
Okay.
pastry section, not within the
All
barbecue.
right.
So,
All right.
okay. So with that,
Cool.
Josh, thank you very much again. And for folks that are on the line, thank you for tuning in to this session of HUPO's Proteome Talks. And until the next session, this is Henry Rodriguez saying stay safe and take care, everyone.
All
Bye-bye.
right. Take care, everybody.